Welcome everyone to this week's edition of the EV Journal Club. My name is Kenneth Whitwer. I'm at Johns Hopkins University, and I'm coming to you today from a very bright and snowy Baltimore. So welcome to all who are joining us from around the world. It's my pleasure today to introduce Naveed Akbar, who is a senior postdoctoral scientist and clinical lecturer in cardiovascular medicine at University of Oxford in the UK. So Naveed, thanks so much for joining us and sharing your work.
Thank you very much, Ken. Thank you for the invitation and thank you all for joining. Today, I'll share with you data on how the injured myocardium mobilizes neutrophils from the spleen, and it induces transcriptional activation of these cells as they're en route to the injured myocardium in the peripheral blood, so before they get to the heart. The majority of the data I'll share with you is unpublished, but it's part of this preprint, and I'm keen to hear your views. So the etiology, the background behind acute myocardial infarction and heart attacks is well known. Risk factors such as a sedentary lifestyle, smoking, or diets which are high in cholesterol, and genetic factors such as ethnicity influence our overall risk for myocardial infarction. But irrespective of the underlying pathology, it's atherosclerosis, which leads to the event. Fatty deposits within coronary arteries, which over decades become very active with the recruitment of immune cells, and they develop these atherosclerotic plaques, which are very, very active. So a rupture or blockage in one of these coronary arteries results in myocardial infarction, where you sustain localized ischemic damage to the heart. And the primary therapy here is percutaneous coronary intervention, where a treating physician might remove the clot and implant a stent. This is very effective, and it's partly responsible for the decline in the number of deaths that we've seen with MI over the past few decades. But once the heart is injured, in the subsequent hours and days, it sustains secondary inflammatory mediated damage. In the immediate hours following myocardial infarction, you get a robust immune response, which results in elevated blood neutrophil counts. These are present in patients when they arrive at the hospital, and they're prognostic. So patients with very, very high levels of immune cells in their blood, as they come into the hospital, this is associated with poor outcome. What I mean by this is increased mortality, a bigger injury size, and poor cardiac function upon recovery. Today, I'm going to focus on a subset of peripheral blood cells called neutrophils. So after a heart attack, substantial numbers of neutrophils infiltrate the injured heart in the immediate hour. Deletion of immune cells or inhibition of recruitment to the heart lowers the final injury size, but doesn't always result in better function. And that's because immune cells mediate both aspects of further injury and repair following a heart attack. Once inside the myocardium, neutrophils will activate, they'll degranulate, and they'll prime the inflammatory environment for the subsequent arrival of monocytes, which are well studied in the setting of heart injury and repair. Neutrophils are the most abundant immune cell in peripheral blood and in the injured heart 24 hours following injury. And they're the earliest immune cell to arrive, and their number in the peripheral blood correlates with how big the infarct is in patients. But how do neutrophils get recruited to the injured heart? How do their gene expression profiles get altered? And where do the cells come from? These questions remain largely unknown, but a better understanding of these processes in the immediate hours following injury may allow us to target the first cells which infiltrate the injured heart to salvage the myocardium from inflammatory damage. So we hypothesize that neutrophils are mobilized to the peripheral blood from reserves such as the spleen or the bone marrow, and that the injured heart is able to signal this mobilization. We further hypothesize that analysis of neutrophil gene expression profiles will allow us to identify pathways and processes that are amenable to therapeutic intervention. So the data I'm going to share with you today is a combination of human patient data, cell culture data, and then some rodent experiments as well. Bear with me, I just need to move this out of the way. In Oxford, we benefit from the Oxford Acute Myocardial Infarction Study, where patients presenting with myocardial injury are highly characterized with respective MRI imaging and serial blood sampling. So in agreement with previous studies, we find that the plasma neutrophil number at the time of presentation, when the patient comes into hospital, is associated with a larger infarct and larger scar 6 months following the myocardial infarction. In these graphs, you can see the number of neutrophils from the patient as they arrive on the y-axis, and then we use MRI to look at the amount of edema or water in the heart, which is a surrogate for the injury size. 6 months later, we can use late gadolinium enhancement, and in both measurements, which are the same patients, we find that neutrophils in the blood result in a larger infarct. The rapid elevation in peripheral blood neutrophils in the immediate hours following MI raises the question of how neutrophils are mobilized and where they're mobilized from. To investigate whether the spleen or bone marrow mobilizes neutrophils after injury, because these are reserves, I've used a mouse model of acute myocardial infarction here, where we ligate the coronary artery, we allow the animals to recover, and then we harvest tissues of interest, such as the blood, the spleen, the bone marrow, and the heart, and we can determine neutrophil populations by flow cytometry. We find that there's a rapid increase in peripheral blood neutrophils, a simultaneous reduction in splenic neutrophil number, but at this very early time point, we see no alteration in the bone marrow. So this is pretty exciting. It's not been demonstrated before that neutrophils mobilize from the spleen following sterile injury, following a heart attack. And we think that's because traditionally research groups like us who are interested in immune cell activation and immune cell kinetics after injury don't look at these very early time points. But we hypothesize that maybe the mobilized peripheral blood neutrophils would undergo alterations in their gene expression before recruitment to the myocardium, similar to what we have previously observed in monocytes. To determine this, I used RNA sequencing. Here we used N-STEMI control patients and STEMI patients at the time of presentation. And then we followed the same patients up 1 month later to obtain a matched control sample. STEMI patients show alterations typical of a myocardial infarction on an ECG. They'll also show a rise in cardiac troponin. N-STEMI patients may have a normal ECG and normal troponin levels, but upon an invasive clinical angiogram, the treating physician may find a blocked vessel and treat with a stent, whereas STEMI patients are always immediately treated with a stent to open up the blockage. So using unbiased analysis, such as this PCA plot, we find that STEMI patients group together and that there's a clear delineation between the response at the time of injury versus the response at follow-up. Looking more closely at the absolute number of genes that change in each group, which are highlighted in red, we find that over 900 genes are altered in the STEMI group, which is surprising because neutrophils are regarded as cells that are transcriptionally poor. Employing further bioinformatics to determine whether the genes that change are favoring particular pathways, we find that there's strong representation of inflammatory pathways with significant enrichment for pro-inflammatory TNF-alpha and interleukin-6. This changes the way that we currently think of neutrophils because they're not just responding to the local environment in the myocardium, but they have prior priming or programming. And this opens the possibility of targeting the cells therapeutically to alter their function and maybe promote regeneration and repair. But how does the injured heart communicate with the spleen to induce the mobilization and the transcriptional activation? Whilst the soluble factor from the myocardium could mediate the response, the concentration gradient is inverted. It's possibly very high in the heart and it's lower in peripheral blood in the spleen. But even if you look at the coronary sinus, so that's undiluted coronary blood, the elevation in chemokines following PCI is around 0.1-fold. But neutrophil numbers in peripheral blood are already elevated. And if you look back at the studies conducted in the 60s, the 70s, and the 80s that show neutrophil mobilization from the bone marrow, they used intravenous chemokine injections that were several orders of magnitude, and by this I mean 400 to 600-fold greater than the very modest elevations that we can detect within the heart of patients following PCI. This suggests that other mechanisms might be responsible for the immediate mobilization of neutrophils following myocardial infarction. And we hypothesized that extracellular vesicles, which are these membrane-enclosed envelopes, carry proteins on their surface, RNAs within their spheres, could mediate this effect. And I've shown previously that extracellular vesicles could mobilize monocytes from the spleen to peripheral blood following a heart attack. I'm aware that there's some non-EV researchers on this call, and I'll briefly explain some of the methodology that I've used. So EVs is a broad term, refers to three main populations of extracellular vesicles: apoptotic bodies, mid-sized microvesicles, and the smallest are called exosomes. But there's a significant overlap between these populations, and it's difficult to determine the subpopulations accurately, especially in highly heterogeneous matrices, such as the plasma, which is what I'm using here. So extracellular vesicles can be isolated from biological fluids, including plasma, by using ultracentrifugation, which is what I've used here, where you initially pellet off cells and debris, and then using greater force, you can pull down microvesicles and then the smaller exosomes. The data I'll share with you today is a combined population of microvesicles and exosomes, which I'll call extracellular vesicles. The isolated EVs, they were washed with PBS, and I've utilized them in the studies I'll describe. I'll cover the characterization as we progress through that data. I have confirmed these findings using other methods, such as size exclusion chromatography, coupled with ultracentrifugation, and we're in the process of repeating some of this analysis using immunoaffinity capture against tetraspanins and integrins. But in the interest of time, I won't go into the benefits and the limitations of each of those methods. So we tested the hypothesis that extracellular vesicles mediate the neutrophil response in MI. I isolated plasma EVs at the time of presentation, and then 6 months later in the same patient to serve as a control. So the patients presenting with MI have significantly more EVs at the time of injury versus the 6-month control measurement, but they show similar size profiles. And the concentration of plasma EVs is on the y-axis here, and on the x-axis, you see the size of the particle. We looked at these particles using TEM. They show this cup-shaped morphology or circular morphology, and we have looked at the tetraspanin CD9 and CD63. I've done a more detailed look at the plasma EV fraction to show that they're absent of platelets, cellular contaminants, such as mitochondria and histones, but I want to focus on the clinical data here, but you can refer to it in the preprint. So plasma EV number at the time of presentation, so when the patient comes into the hospital, correlates with the extent of myocardial injury. That's both the amount of edema measurements of the T2 weighted MRI and the size of the scar 6 months later, determined by late gadolinium enhancement. So these are the same patients that I showed you earlier. A larger EV count results in a larger accumulation of edema or a larger scar. We also found a highly significant relationship between the plasma EV number at the time of presentation and the number of neutrophils in the blood. Now, although this is just an association, it raises the question that EVs may mobilize or interact with neutrophils following myocardial injury. So EVs are elevated following myocardial infarction, as are neutrophils, and both of them correlate with the extent of injury in patients. But where are the EVs coming from? And EVs contain bioactive substances such as RNAs. Are these altered following myocardial injury? To determine this, or to determine whether we see alterations in the microRNA content of EVs following MI, I isolated plasma EVs at the time of presentation and then 6 months later to serve as a control. And we screened over 700 microRNAs using a reverse transcription qPCR array. What we find is that 12 microRNAs are significantly enriched in plasma EVs at the time of injury. But this is an oversimplification of the model that we use. Alongside screening the 700 microRNAs, we utilized the regression model, where we take EV density and patient characteristics such as sex, the size of the infarct, into consideration to determine which EV microRNAs are changing significantly following injury. Now, there are many cell types within the heart that can produce extracellular vesicles, but all of these microRNAs are present in endothelial cells, and two in particular, microRNA 1263P and microRNA 1265P, are endothelial cell associated or endothelial cell specific. MicroRNA 126 is of particular interest to us because endothelial cells are a highly plausible source for the potential generation of a signal in the injured myocardium to recruit immune cells from a distal reserve. They're in very close proximity to the infarcted myocardium. They're bathed within the ischemic environment, and they may produce and release EVs to signal immune cell recruitment. Therefore, to examine whether endothelial cells produce EVs, I've used a model of human endothelial cell activation in vitro and then isolated the EVs. Here I cultured primary human endothelial cells. We stimulated with a pro-inflammatory factor, so recombinant TNF alpha, and we isolated the EVs using ultracentrifugation with washing. We can confirm that the endothelial cells are activated by looking at the presence of VCAM1 using an ELISA, and we find that there's significant enrichment following the stimulation. The human endothelial cells produce and release EVs under basal conditions, but they release significantly more following stimulation with the pro-inflammatory TNF alpha. And although the size profile of the EVs is similar with a potential increase in small EVs, they show a typical morphology and are positive for CD9. In these particular preparations, I've used a sham media control to account for the bovine-derived EVs in the culture media. This is an important control because bovine EVs carry bioactive materials such as microRNAs, which show sequence homology to the human and the mouse. From these preparations, I wanted to know whether we would potentially see any differences in the microRNA 126 content following inflammatory stimulation. So I isolated the EVs from similar preparations, and we looked at this using RT-qPCR. And what we find is an enrichment for microRNA 126 from endothelial cells following inflammatory stimulation, supporting our clinical observations. MicroRNA 126 is enriched in activated endothelial cell-derived EVs, and it's present in the plasma EVs from our patients at the time of injury. But does it have any effect on neutrophil biology? To determine this, and because microRNAs control gene expression, we have used in silico bioinformatics. By looking at the seed sequence of the microRNA that we're interested in, so here it's microRNA 126, we can look at the putative or the predicted targets. And I wanted to know whether there was similarity between the human and the mouse, because this could allow us to do some mechanistic studies further on. Using in silico techniques, we take our microRNA of interest, we use these curated databases. Here I'm using 3 different databases to increase the stringency of our analysis. And then, I've skipped ahead. And what we have done is we have looked at commonality between all of the protein coding genes between the mouse and the human. And what we find is that there's a significant overlap of 30% between the 2 species. We further assessed commonality between the human and the mouse microRNA-related messenger RNA lists using gene ontology to determine whether the predicted targets of 126 were similar in the human and the mouse when they centered around biological functions. So using a similar approach, we're using a Fisher's exact test. We assess commonality between the microRNA 126 messenger RNAs and the pathways that they target between the mouse and the human. We see an overlap of about 40%, including gene ontology terms for the regulation of cell motility and cell migration. But this doesn't tell us that much about neutrophils. It tells us that there's a nice overlap between the species. Using a similar approach, I wanted to determine whether the microRNA 126 targets were present in gene ontology terms for neutrophil functions. So we looked at the predicted targets again, so from the human, the mouse, and the ones which appear in both species. And we downloaded all of the neutrophil terms in gene ontology, and we looked for overlap using the Fisher's exact test. What we find is that microRNA 126 targets are overrepresented in neutrophil gene ontology terms for migration and for chemotaxis, but not for other terms such as neutrophil clearance and neutrophil-mediated killing of a fungus. This is exciting, it supports our hypothesis, but do microRNA 126 targets change in neutrophils following AMI? To determine this, or determine whether the microRNA 126 targets are enriched in neutrophils following injury, following a heart attack, I looked at the STEMI patients that I showed you earlier, where we're seeing the 900 genes that changed, and we compared the list of differentially enriched genes, so the ones that are going up or down, in the neutrophils to the predicted targets or the putative targets of microRNA 126. And we find significant overlap with the human predicted targets. Building on this, I wanted to know whether we can get similar responses in the mouse, so we can then mechanistically look at this in subsequent studies. So Clement and his team have recently published this paper where they've performed MI in the mouse. They've then used single cell RNA sequencing of blood neutrophils, and they show that there's heterogeneity in the blood neutrophils following myocardial infarction, and that they can be grouped or clustered here in this unbiased map where we're showing 6 clusters. Using a similar method, the Fisher's exact test, we find that there's a similarity in the genes that change between the mouse and the human following myocardial infarction, and we find that there's enrichment for the microRNA 126 targets within specific neutrophil clusters. We've tested whether targeting microRNA 126 using an integument in the mouse lowers infarct size, and we see a significant reduction in infarct size. We've done that in collaboration with Catherine Moore's group in New York, but I'm not going to share that data with you today. Instead, I'm going to focus on the EVU response. So endothelial cell-associated microRNA targets are represented in neutrophil transcriptomes at the time of injury in the human and the mouse. Targeting microRNA 126 with an integument lowers infarct size, but EVs will also carry proteins on their surface from their parent cell, and endothelial cell integrins are well described. Endothelial cell integrins such as CD1, ICAM, VCAM1 are expressed in endothelial cells, and they show significant enrichment following inflammatory stimulation, such as the simulations I showed you earlier with TNF-alpha with an enrichment for VCAM1. To determine whether endothelial cell integrins are present on plasma EVs at the time of injury, I isolated plasma EVs from my patients and the mouse model of myocardial infarction, and we found enrichment for VCAM1. But what about endothelial cells? If you do a similar assay to what I showed you earlier, you take mouse or human endothelial cells in culture, you stimulate them with TNF-alpha, you harvest the EVs, we find that there's significant enrichment for VCAM1 in vivo and in vitro, whether you're human or whether you're a mouse. But one of my concerns at this time was that the VCAM1 signal was generated by soluble VCAM that was co-isolated with the EVs, as most isolation methods have some inherent contamination, and ultracentrifugation isn't exempt from that. To show that the VCAM1 was on the surface of the EVs, I isolated plasma EVs. We took magnetic beads of iron oxide, we conjugated anti-VCAM1 antibodies to the surface, and we used an IgG control, so similar to performing an IP. And we've previously used this method of fabricating magnetic particles of iron oxide for imaging. What you can do is you can incubate your isolated EVs with your immunogenic magnetic beads. You can then enrich your EVs by washing them, embed them into agar, section them, and then look at them using TEM. And what you find is that there are VCAM1 positive EVs decorated around the magnetic bead. If you look at the IgG, it's completely clear. And our PhD student, Dan, who's on the call right now, is in the process of applying this enrichment approach across our clinical cohorts. Endothelial cell-derived EVs are enriched in the blood following myocardial infarction, but do they localize to the spleen, and could they possibly be mediating this neutrophil response that we're observing? It's difficult to determine the biodistribution of EVs in vivo because they're so small, but I've used a labeling technique that allows us to assess tissue distribution in the mouse. So I've kept the species of EVs for these studies the same. So similar to the human experiment that I showed you earlier, we take some mouse endothelial cells, we stimulate them with TNF-alpha. We can confirm that the cells are activated using this VCAM1 ELISA. The mouse endothelial cells produce EVs under basal conditions. They produce significantly more following the inflammatory stimulation. We're able to show that they have typical morphology, and we've looked at the EV markers, which I'm not going to show here, but they're available in the preprint. To allow the quantitative tracing of these mouse endothelial cell-derived EVs in vivo, we've taken a preparation and I've transfected them with the non-mammalian microRNA miR39, which is not present in the mouse and it belongs to the worm C. elegans. What you can do is you can transfect your EVs, wash them, and then inject them IV into your mouse, harvest tissues of interest, and you can use RT-qPCR to determine where they've gone. We find that the EVs are delivered to the spleen within minutes, and they remain detectable for up to 24 hours. This technique has some inherent limitations because you can't be certain that it's still the EVs that we're detecting at all of these time points or whether it's just the transcript. But it does allow us to determine that we get endothelial cell-derived EVs delivered to the spleen. Endothelial cell-derived EVs are enriched in the blood following myocardial infarction in the mouse and in the human, and there appears to be control of neutrophil activation by the microRNA 126. But once they localize to the spleen, do they have any functional effects there? To determine this, I wanted to see if there's any change in inflammatory gene expression or any chemokine signaling pathways that may coordinate neutrophil movement or neutrophil mobilization. So what we've done here is we've taken the preparation of inflammatory activated endothelial cell-derived EVs, injected them into otherwise healthy animals, harvested the spleen, and then done an RT-qPCR for gene expression or done a protein array. What we find is that the expression of CXCL1, CXCR4, interleukin-1 beta, and IL-6 rapidly increase post-injection. And this time point is important because it matches with the time point of where we observe the neutrophil mobilization from the spleen in the animal model. And because microRNAs can silence gene expression, we wanted to know whether delivery of the endothelial cell-derived EVs rich in the microRNA 126 could influence protein expression. Used a similar approach here, but we're taking the spleen, we're homogenizing it, exposing it to a chemokine array, and we find significant reductions in 6 chemokines, including targets of 126, which is CXCL12. When we normally think of chemokines, we think of their expression as increasing or rising upon an inflammatory stimulation, but CXCL12 is highly expressed in the spleen and in the bone marrow. And what it does is it retains neutrophils in these reserves. MicroRNA 126 can downregulate CXCL12, and lowered expression of CXCL12 induces neutrophil mobilization to the blood. So informed by this and the previous bioinformatics, I wanted to test whether endothelial cell-derived EVs could mobilize splenic neutrophils. So we injected otherwise healthy mice with a preparation of inflammatory-derived EVs. We harvested the tissues of interest, and we looked at neutrophil populations using flow cytometry. What we find in these otherwise healthy animals is that there's a rapid and substantial increase in peripheral blood neutrophils and a simultaneous reduction in neutrophil number. So this supports our hypothesis that endothelial cell-derived EVs mediate neutrophil mobilization from the spleen, what mediates the effect and can we therapeutically target this? So it's well known that immune cells can be captured to sites of inflammation, by the expression of VCAM1. And the receptor VLA4 is found on immune cells such as neutrophils. But we find that VCAM1 is enriched on EVs in the peripheral blood at the time of injury, and we hypothesize that the inverse might be also true, where plasma-liberated endothelial cell-derived EVs rich in VCAM1 could mediate neutrophil mobilization from the spleen. To test this hypothesis, I CRISPR-edited VCAM1 from some mouse endothelial cells. So upon application of TNF-alpha, they no longer express VCAM1, unlike the wild-type cells. I've characterized the EVs from these knockout cells, which are in the preprint, but I'm not going to share that data with you now. Instead, I'd like to just focus on some of the functional experiments that I've done. So we hypothesize that the VCAM1 may be necessary for delivery of the endothelial cell-derived EVs to the spleen, or for the functional effects that we're seeing, the neutrophil mobilization, the change in the gene expression. To determine this, or to determine whether the VCAM1 was necessary, we took otherwise healthy animals and we injected them with a preparation of the wild-type inflammatory activated VCAM1 rich endothelial cell-derived EVs, or the knockout, at the same concentration. And then 2 hours post-injection, I harvested tissues of interest. And what we find is that the VCAM1 knockouts failed to mobilize splenic neutrophils to peripheral blood following the IV injection, unlike the wild-type VCAM1 rich endothelial cell-derived EVs. I did a similar experiment to determine whether we're still seeing any alterations in the gene expression that may be responsible for the mobilization. And we find that the VCAM1 knockouts do not mediate the changes in gene expression that we saw with the wild type, including for the targets of 126, CXCL1, interleukin-1 beta, and IL-6. Thereby, we have a pathway now where we're demonstrating where ischemic tissue can liberate endothelial cell-derived EVs, decorated in a VCAM1 integrin, rich in a microRNA 126. They can localize to the spleen, and they can mediate chemokine regulation and the mobilization of neutrophils. So in summary, acute myocardial infarction induces a peripheral neutrophil response that correlates with the extent of myocardial injury. Plasma EV number is elevated following myocardial infarction and correlates with injury size and the number of neutrophils in the peripheral blood. Plasma EVs are enriched for endothelial-associated microRNA 126 and endothelial cell integrin VCAM1 following myocardial infarction. These can localize to the spleen. Here they can down-regulate chemokines, induce alterations in gene expression, and induce splenic neutrophil mobilization, which is dependent on VCAM1. Neutrophils are activated before they get to the site of injury, so in peripheral blood, and they show enrichment for microRNA 126, targeting in an animal model microRNA 126 with an integument, you can lower infarct size, and therapeutically targeting neutrophil transcriptomes to potentially perturb the inflammatory effects might be a novel therapeutic avenue to salvage the myocardium following sterile injury. Just want to say thank you for your time. Thank you for your attention. Thank you to Robin, who's our PI, head of our laboratory. Thank you to all the patients and researchers who are involved in Oxami, which allows us to get access to these clinical samples. Our funders, the British Heart Foundation, the Centre for Research Excellence in Oxford, the many collaborators have enabled me to undertake this work. And if you have any comments or questions, I'd love to hear them.
Well, thank you so much, Naveed. I really enjoyed your presentation. It was nicely delivered and very clear. We do have some questions that are coming in the chat box, but I wanted to start out with a couple of questions of my own. And this comes from somebody who is not at all an expert in myocardial infarction, so please forgive me if this is a bit naive. But what else is known about biological particles just after MI? I mean, are there other, like I'm thinking about lipoproteins in particular, are there changes to the lipoproteins also following MI?
Changes to the lipoprotein profile after MI. We have done one particular study, but the study was done in patients who were presenting with acute coronary syndrome, so chest pain, And we were looking at a group of patients who were statin naive and then giving them statin therapy after that. So yes, some patients that are coming into the clinic are on lipid lowering therapies, some of them aren't. We haven't looked specifically in patients where that is controlled to see whether we just see changes in the LDL, the HDL ratio. But if we look at preparation of plasma EVs that don't rely on the densities of the ultracentrifugation or the size of the particle, such as size exclusion chromatography, we see similar effects. So if you use immunoaffinity capture against the trispanins, and then we look at similar time points, we're still seeing similar effects in terms of the EV response.
Okay, good. All right, thank you. Let me see here. I'm going to allow unmuting now. So people can ask their questions. Let's start with Phil Askinase. Phil, you have a few questions here that I think are very interesting. Go ahead.
Hello, Naveed.
Hi.
So the first thing is, how early in an MI, and what's the time course of the EV effect, and how does it compare to troponin?
So in this, the patients that I've shared with you today, the median time to presentation was around 3 hours, but that varies considerably. Some patients may have had chest pain 12 hours before they went to bed, and it's sometimes a considerable amount of time before they pick up the phone and call an ambulance. And so from the clinical cohort, it varies quite considerably. From the animal model, we have looked at 24 hours post-infarct, and we know that we see this rise of plasma extracellular vesicles. It's still sustained at 24 hours, but if we look at 7 days or 14 days after infarct, it's come back down to baseline. I'm
talking, what about an hour? Real early.
So I'm in the process of doing that now. So using the Oxami cohort, the patients were in hospital for a couple of days, and then we follow them up for 6 months later. So we've taken the same patients that I've described today, and we've looked at them at 6 different time points. And I'm in the process of looking at 20 different EV markers to see how they map or they change as the infarct evolves.
You can look at the mice in 1 hour.
We haven't done that yet.
Well, that's going to tell you early. I'm wondering about that some of the changes, the spleen might be indirect, that the target in the spleen is a monocyte macrophage, and then that secondarily affects the subsequent release.
So whether the accumulation of the endothelial cell-derived EVs is non-specific and that it exerts an effect anyway? Was that your question?
No, as to whether the effect in the spleen is direct or indirect via the monocyte macrophages in the spleen.
So it's a great question. So in the model where we induce a rapid infarct, and then 2 hours later, we look at the cell populations, the neutrophils mobilize very, very quickly, but the monocytes haven't left yet. But if we do the exosome experiment where we take the inflammatory activated endothelial cell-derived EVs and we inject them into naive animals, we get mobilization of both the monocytes and the neutrophils. So potentially in vivo, we need data to support this, but I'm currently thinking that there might be different waves of endothelial cell-derived EVs, one that maybe coordinates neutrophil mobilization, which is very early, and maybe a second wave or change in EV composition, which coordinates the monocyte mobilization.
So this sort of thing happens in injuries in other organs. In a stroke, the same sort of thing happens. How's it different?
So we've done the studies in stroke, and we've done some adoptive transfer studies in stroke. We haven't looked at the splenic immune response, but we have looked at the acute phase response and sickness behavior. So if you induce injury in the brain, we've shown that there's communication of EVs across the brain to the liver, where they're accumulating macrophages and they induce the acute phase response. And if you delete or you deplete macrophages using clodronate, liposomes, you can abrogate that signaling. that behavior.
Well, it's all fantastic. Very, very well presented, as Ken said.
Thank you, Phil. Thanks for those questions. I'm going to move to another question that had to do with this clinical cohort. This is from the Oriel College Junior Deans. In that case, then, in the clinical cohort, is there any correlation between the time sample that was collected post-infarct and the EV concentration that you measured?
Is there any correlation between the sample that was collected post-infarct? So
the time, I guess time, is there any trends over time? So this is related to Phil's question for sure.
Yes. So what's happening in those very few immediate hours of EV kinetics and immune kinetics? Do they completely match up? We don't have that data at the moment, but I'm in the process of, we have sent that for analysis. So what we'll have is patient comes in straight to the hospital. We're able to take a blood sample before they receive any clinical therapy. Then we have a blood sample that comes immediately after PCI. So as soon as the stent has gone in, we can then get another sample 24 hours, 48 hours after, just as they're discharged, and then 6 months later. And so we'll be able to do some time resolved kinetics of the EVs and maybe the constituent parts of the EVs, the protein, the lipid, the potential RNA contained within them.
Good. All right. So let's go now to James Drummond. James, would you like to ask your question about VCAM?
Yeah. So I thought your knockout experiments were very nice. I just, as sort of an adjunct to that, if you could block the VCAM with an antibody, either directly on the EVs prior to injection, or just add it to your mouse models, would that then, what would you expect to see with that if you tried that?
So surprisingly, no one has ever done this. No one has actually used a VCAM1 antibody in a mouse model of myocardial infarction. And we have done an experiment along that line, but the dosage and the timing is quite complicated. So we don't know when the absolute rise in the VCAM1 positive EVs, when does that happen? Is the antibody going to be around a sufficient quantity to mop up that signal? We thought about doing, or I thought about doing the pre-treatment experiment that you're describing. So you take your isolated EVs, pre-incubate them. But at that time, I was really concerned about shoving stuff onto the EVs that would change their behavior, change their kinetics. And so in our setup, actually, we have endothelial cells that produce EVs under basal conditions, which don't have as much VCAM1. You inflammatory stimulate them, they have a lot more. And then we were able to the CRISPR-Cas9 editing reasonably simply to generate that knockout.
Okay, thank you.
So Naveed, with your VCAM, can you distinguish between there just being more VCAM per EV, or is it that you have more EVs with VCAM?
To be determined. Both are fascinating questions. And so are the cells within the myocardium producing more EVs, which are loaded with more VCAM1, or a combination of both? We don't know at the moment.
Good. Okay. Hojian Kai, you have another question about VCAM. Go ahead.
This is just a follow-up with probably the VCAM theory. So the question is how this specificity of VCAM can target a particular organ. I mean, it could go to multiple organs, and then it triggers this kind of try to recover signals. So the question is how specificity of this VCAM target organs.
That's a great question. And so we're in the process of, we've just done that experiment. So if you take our labeling technique, where we are able to introduce the C. elegans microRNA, then we can do this quantitative tracing in the mouse. But instead of just taking the spleen, you take every single tissue you can in the mouse, and then you do the wild type in the VCAM1 knockout. But the early data that we have, which is already in the preprint, we don't seem to affect the accumulation pattern by deleting the VCAM1. The EVs using our method accumulate at the same rate. But what's really exciting about that is that the clearance to the spleen is no difference between the knockout and the VCAM1, but the VCAM1 or the loss of the VCAM1 still results in a functional loss. We still don't see the mobilization. We still don't see those gene expression changes that we see with the wild type.
Good, well done.
Thank you. Ellen Millman, you have a question about what's happening elsewhere.
Hello, thank you for taking this question. Your data is lovely, but I'm interested in if you're seeing an infarct in the heart, how much of that inflammation signal is being spread to other parts of the body, particularly the lungs and kidney. Does the infarct size generate a bigger generalized inflammation signal?
So what we know from rodent experiments that we have done and published, that if you have a myocardial infarction, that you will see upregulation of VCAM1, activation of endothelial beds in other organs, in particular the kidney. And so we haven't looked at every other organ to see how pan-activation of endothelial cells is taking place following myocardial infarction. But that's really interesting, is all of the contribution that we're detecting in the patients and in the rodent model in peripheral blood, is it all coming from the heart, or do we see contributions from other vascular beds?
And have you looked for neutrophils in those other beds?
No, that's another pending experiment.
Common problem.
Thank you.
Thank you, Ellen. So now a question from Yamini Nanduri. Yamini, you have some methods questions here.
Yeah, sorry, it's more technical question. But I really like your C. elegans miRNA-based tracking of EVs. Oh my God, that's very, very interesting to track EVs after transplantation. So my question is more a technical one with that. How do you isolate miRNA from the ultracentrifugation derived EVs? Because I am doing ultracentrifugation of EVs, we do see good characterization with EM and particle size measurements and functionality in vivo in vitro. But when I'm trying to isolate miRNA, I'm having a lot of issues with the quality. So I'm just curious which method you use.
So the full details are in the methods of this preprint, but we're using reasonably standard techniques, using a kit from QIAGEN called Marina, allows you to do total RNA extraction. For this, the data that I've shown you today, we did an RT-qPCR by a company that doesn't exist anymore called Exequon. They merged with QIAGEN, so the technology was transferred over, and so that's why I use that particular kit there. But I have also done this, just doing the bioanalyzer, we get good quality. And then by doing the Affymetrix microRNA array, we see good signal. So taking patients at time of injury versus follow-up, we're seeing differential patterns of activation there.
Yeah, I would just add that if you are having problems, just make sure that your isolation method is allowing you to capture those really small RNAs, because there are some RNA isolation kits that are really just focusing on the ones that are above 200 nucleotides or so.
Actually, that is very interesting, because when I'm doing a total RNA and when I measure it on bioanalyzer, I see microRNA, but I also see these really long RNAs, which kind of scary for me, as I'm not sure that EVs would have that long RNAs that are more than 1,000 BP. So I was just worried that is there any contamination due to ultracentrifugation? That's where I'm stuck. Yeah. And do you use ultracentrifugation also for isolating EVs from the mouse plasma?
Have done, started with, as did many of you, we all started with ultracentrifugation, various rounds, doing various clearance, moved that on to doing size exclusion, then size exclusion with ultracentrifugation, and now we're trying to move that on further to allow us to handle more and more samples.
Yeah, great, yeah. Thank you, I would definitely refer to the paper.
Thank you.
Thank you.
Thank you. Now, Marina Ramirez Alvarado from Mayo Clinic. You have a comment and a question.
Yeah, so my comment is that we've been doing isolation of urinary EVs in renal amyloidosis patients. And there we are seeing a decrease in the numbers compared to normal controls or other plasma cell disorders. And we are wanting to move on to cardiac amyloidosis EVs that we will be characterizing from plasma. And we were thinking that we were going to see the same thing, but your data is inspiring me to broaden my theory, because perhaps there's going to be some similar damaged response that will increase the number of EVs. That's the comment. Question is, and I understand that because you are working with a mouse model, perhaps the way you induce the myocardial infarction may not be the same as humans, but have you thought about following what is happening with these EVs, the atherosclerotic plaque, neutrophil enrichment with the atherosclerotic plaque, and sort of that milieu of environments that are created pre and post myocardial infarction in the context of these EVs, the studies that you are doing?
Thank you for both your comment and for your question. Just on the basis of your comment, I think as scientists, we always get very excited when we see something goes up, right? We give it causality, we give it a bit of a metaphor. In some situations, though, I have seen in rodent preparations where we're not seeing an increase in a pathological state, we're seeing a decrease. And that opens up interesting questions in its own right. Is it that particular organ that's affected? Is clearance maybe enhanced? Are they being lost somewhere else? All of those questions are open as far as I'm aware. Your second question is completely relevant and completely true. So here we're taking otherwise really healthy endothelial cells in culture, we're giving them an inflammatory factor because it's an accepted model of endothelial cell activation. But in our patients, certainly, they're going to have underlying vascular disease, endothelial dysfunction. There might be dyslipidemic, there might be hypertensive on top. How do those factors contribute to the overall EV response? Do they contribute is the first question. And are they additive? Do you get more and more EVs? Do you see more and more enrichment, or does it max out? As far as I'm aware, all of those are unanswered questions. We don't know. And we haven't at the moment moved into those complex in vitro setups to test whether they have similar or different functional effects.
And I'm going to ask a question here for Yvonne Couch because of microphone issues. So she asks, we know that neutrophils produce EV trails when they're activated. Could circulating monocytes be doing the same thing in vivo when you are doing your IV injection?
So, hi, Yvonne. I know Yvonne very well. We're based in the same hospital. The stroke work I mentioned earlier, Yvonne led on that. In the in vivo injections, the animals are otherwise completely healthy. They're only exposed to the EVs for 2 hours. What we see is that the immune cells are leaving the spleen, they're entering peripheral blood, but we haven't done any further characterization on that at the moment. We are seeing some differences in gene expression and protein expression, which map the mobilization signature, but we haven't gone on to then question what happens to those immune cells once they're mobilized into peripheral blood. Are they sticking around? Are they activating? Do they go to a different organ? That's an open question at the moment.
All right, very good. Now, Augustine, you have a question here about the types of EVs that might be present. And in fact, I also see that Maxime is asking something similar. So go ahead, Augustine.
Yes, thank you, Kenneth. Thank you very much, Naveed. Very nice presentation and very interesting work. I was more wondering about when you mentioned plasma EVs, and I was wondering about what is this composition of EVs? What do you see any major groups of EVs from a specific type of cell in plasma?
So I completely agree with your comment. So the peripheral blood is a heterogeneous swamp of EVs derived from many tissues from many different sources. Following the myocardial infarction observation that we see an increase in extracellular vesicles, we did the microRNA experiment where we looked at the RNAs enclosed within those EVs at the time of presentation and 6 months later. We treated that in an unbiased way. And so when we found that the 12 microRNAs were changing, that's when we were starting to think about what could be the contributing cell type here. And then microRNA 1263P and 5P both popped up. And then if you just do a qPCR experiment looking at all 12 microRNAs, they're all expressed in endothelial cells. And so we're looking at MI, it's a vascular disease. We know that the vascular endothelium is very important in the capture of immune cells. That's where we have focused our energies. But if you do similar experiments to those that I've described to you today, where we take cells in culture, we expose them to an inflammatory factor like the TNF-alpha, if you do that with cardiomyocytes, we don't see the same response. But other people have reported if you modulate the concentration of glucose or if you give it oxygen hypoxia response, cardiomyocytes do release some amount of EVs or change the capacity in which they release EVs. But we see this very, very robust response in the human peripheral plasma, mouse peripheral plasma. We see the enrichment for the VCAM1. VCAM1 in otherwise healthy animals, healthy humans, isn't expressed within the vasculature. Yes, maybe some in the bone marrow, some in the spleen. And that's what drew us to an endothelial cell hypothesis. And the data is supportive of that.
Right. And for instance, in plasma of healthy donors, do you have an idea of what are the major subgroups, or maybe, so do you think maybe also endothelial-derived vesicles, but could it be also, I don't know, maybe erythrocyte-derived vesicles or granulocyte-derived vesicles? Any idea about the composition of these vesicles in plasma, for instance, from healthy donors?
So we haven't done a full-on, but many people have, where you're taking a pool of plasma and you're saying this percentage is coming from erythrocytes, this percentage is coming from platelets, this is what we can put down to endothelial cells, because they inevitably rely on one or two markers to pull out their associated cell type. The VCAM1 that I showed you, EV, that was stuck to the bead, that's my own VCAM1 positive EV. So they're in circulation in otherwise healthy people. I'll put myself in that bracket for now. What we also know from the mouse is if we work out how many of the particles are VCAM1 positive, so using the bead isolation, about 30% of them are VCAM1 positive. And that seems to account for the uplift or the increase that we see in the mouse model. We haven't done that yet in the human, but we're in the process of doing it.
Great. Thank you very much.
Thank you.
We have two more questions. One from Tobin Kamet.
Yeah, hi, Naveed. Thanks, Ken, for taking the question. Naveed, a great presentation, really well presented. I have a question because you showed in the VCAM enriched EVs bar graph where p-selectin is elevated, and just wondering if you have or if you plan to look for complement deposition on these EVs, suggesting maybe pre and post stent if you see levels that overwhelm the regulators and actually get deposition. Just curious your thoughts or your intentions.
So what we haven't done is looked at complement specifically, but what we are in the process of doing is taking that very, very highly characterized clinical cohort and pulling out EVs at various time points and then doing proteomics. And so we know from the proteomics that we see, especially if you're working with plasmid EVs, a range of proteins which may or may not be EV associated. And so we'll be able to pick up some of that information there, hopefully.
Yeah, the proteomics would be great, but probably just surface phenotyping by Luminex or something would give you a quick assessment of complement. Great. Thank you.
Interesting question. Thank you. And finally, let's go to Shannon Pendergrast, who has noticed some apparent discrepancies in the literature. Shannon.
Hi, Naveed. Great story. For years, MER126, especially a plasma MER126, has been flagged as a potential biomarker for AMI, but there's some conflicting, there's papers, some saying MER126 plasma goes up, some saying it goes down. Most of these papers are not looking at EVs, they're looking at total plasma. Have you, do you have any comments on that discrepancy in the literature? Maybe your more specific methods have solved this controversy?
Thank you for the question. So yes, there is a number of studies out there which show movement in either ways. And if you look at 126 is widely described in diabetes. I'm not going to comment on those studies. What I will comment on is from the patients that I've described to you today, so these oxami cohort, what we have done is a total plasma. It was a clinical fellow that we had, has looked at the total microRNA profile in plasma, and then in the same patients, we looked at the exosomal fraction. And then the 126 goes in the same direction. So following MI, we're seeing an increase in 126, both in total plasma and in the EV fraction.
Thanks.
And I can't help but follow up on that question with a question of my own, which is, do you think that this miR-126 is just in these EVs because of its presence in the I should say the source cell, or is there some specific packaging that's that you think is happening here?
So I think that's a great question. So we know that 126 is necessary following myocardial infarction. So if you're a mouse and you're deficient in 126, you'll have aberrant angiogenesis, and actually you'll have very, very poor cardiac function if you survive after the experimental model. So we know that 126 is vital. Why is it being loaded into these EVs in this particular way that we're seeing? We don't know, but we do know that once it's there, it's having an effect once it accumulates at other sites, in particular the spleen.
All right, well, I think that brings us to the end of our hour. So thank you again, Naveed, for sharing your work with us. It's been a really nice discussion. I think I've learned a lot. So thanks everybody, and we hope to see you again soon. Take care.